The overall goal of this experiment is to accurately quantify the spatial distribution and extent of neuroinflammation in a mouse model of ischemic stroke using TSPO-PET and MR imaging, and to validate these in vivo findings using ex vivo autoradiography.
I'm really excited to share with you this step-by-step guide for how to use PET imaging to visualize neuroinflammation in a living subject. Here we're using a mouse model of stroke as an example. Now, why might you want to use this technique? There are a couple of reasons. The first is that neuroinflammation, which is just inflammation that occurs in the central nervous system, is thought to be intimately associated and really something that underlies many, many brain diseases, including stroke, but also Alzheimer's disease, multiple sclerosis, Parkinson's, and the list goes on. And so it's incredibly important that we have these techniques that really allow us to study these biological phenomena. Secondly, PET imaging offers a number of advantages over some of the more traditional techniques. For example, most of these traditional techniques rely heavily upon using both human and mouse post-mortem brain tissue. And while they have afforded a number of important insights, these techniques are static by nature, meaning that they can only really tell us information about one moment in time. And since we know that both the brain and the immune system are highly dynamic, it makes sense that we'd want to have a technique that allows us to really capture these molecular processes in living intact systems, so in their native environment in real time. And that's exactly what PET imaging does. Here, we'll show you how we use a specific PET radiotracer, C11DPA713, which is known to bind to the TSPO or translocator protein. We'll show you specifically how we inject an image not only one mouse, but 4 mice.